morning, everyone. I am so delighted to be here this morning. I think this is our, from what I was told, this is like our first event gathering together in person, although we're not all of us here today. I'm just glad that we're able to take this opportunity to be here. But again, for Jody's instruction, I have to read this script out loud before we move on. So this conference is accredited for CME by the Yale Medical School. There are no commercial sponsors of this conference. Disclosures were reviewed and no conflicts of interest were found. Now that is out of the way, again, I am delighted that we're here today, and again, super delighted that Dr. Clayton Yates is able to join us this morning, all the way from warm Alabama to be here today. In reviewing Dr. Yates, there are, it was just too big, I think, for me to be able to summarize it. There's just a couple things I do want to point out about him. He received his undergraduate education from Tuskegee University in 1998, then did his PhD in pathology at University of Pittsburgh in 2005. Thereafter, he did his postdoctoral fellowship with Leland Chung at Emory University. Since then, from 2007, he has been a faculty member at Tuskegee University, where he has risen through the ranks, and he's currently a professor there right now. He has received numerous awards and numerous publications. I'm just going to highlight a couple of the awards that he has won. So he has been on multiple Department of Defense study section for the prostate cancer funding program. He was named one of 100 most inspiring African American scientists in the year 2020. He is a charter member of NCI's Developmental Therapeutics Study Section. He has been featured by the National Cancer Institute for his professional career on the NCI website in 2021, which will be submitted to the Congress and President. So needless to say, he has an illustrious career. He has published 74 papers to date. And again, very impressive list. Let me just put the last three publications by him. Most recent one has been published or has been accepted in Cancer Cell. Just before that, JNCI. And just before that, Journal of Clinical Oncology. So the papers that he's putting out, publications he's putting out, just making huge impacts in our field. He has currently 4 NIH, what I can count, 4 NIH fund grants, and 1 Department of Defense funds, and have like multiple grants in pending or in review right now. So again, super productive with his career. In addition to that, I had a dinner with him last night as I picked him up at the Hartford Airport. And there's a couple things that I learned about it, but I don't want to take up too much time. But there are 2 things about him that I learned yesterday that was not apparent in his CV. First, he recently just took a molecule that he developed in his laboratory and just signed a license agreement last week. So he's truly a translational scientist that's actually taken something from the bench all the way to the clinic, now to the clinical trials. And again, he's the one who did that. But most important of all, what I found out about him last night, again, I've been we've been working for about 2 or 3 years, right? But this is the first time actually meeting Clayton in person, by the way, because of COVID. So what I learned about him is that truly he's a man with character, and I think the humbleness he comes to us with it, I think just I'm very impressed last night. So really looking forward to what he has to share with us today. So title for his talk today is Immuno-Oncology Targets in African American Cancer Patients. Thank you, Dr. Yates, for coming.
Well, I'm so honored to be here, and thank you for inviting me as well, and thank you for reading that off. I am humbled to be here at Yale, and also honored to share some of the work that we have been doing over the years with a number of collaborators. I'd like to start off, this is Tuskegee. It's a small HBCU, and after I trained at all of those places, I did go to Tuskegee, and there wasn't much there, so we built this program from scratch. And so I like to consider myself a builder. And so if you can see that in the work that we assume from now, and the momentum that we gained, I think is we're right on track of starting to understand how we diversify patient population and target people effectively with the right medicine at the right time. So just a few disclosures there. As I mentioned, we did able to take a compound and start to license it, and I'm consulting for several therapeutic companies on particularly how they can start to transition their therapeutics into disparate populations. So we talk about the introduction, right? So we know the number of cases, and everyone in this room is very familiar with, you know, what the rates are as far as cancer death. When we start to look at disparities, particularly in the African American population, we see those rates actually increase compared to other populations. And again, right at the top of that, you know, those lists on top of cancers and prostate and breast, as you can imagine. This is a work published 2018 in JAMA from Franklin Wong, who did his residency at Dana-Farber and now is an assistant professor at UCSF. I work very closely with Franklin. In fact, we just got a PCF grant awarded this week from Movember and the Prostate Cancer Foundation. But what he showed here in SEER data was that, you know, if you look at the progression, is a very busy slide, but if you look at the progression of the disease, Black patients, regardless of whether they're diagnosed early and put on active surveillance, still do worse, right? So there's still a worse outcome, whether they're, you know, early stage or late stage, which suggests that there is some biological factor that's underpinning the aggressiveness of the disease. And if we take that, it's not just in the US, right? So this is a worldwide phenomenon or issue. You know, we'll talk about our efforts in that as well. So if you look at other countries, particularly in, you know, the African countries or patients who have West African or African ancestry, we see those rates really increase, right? And we see Black males, you know, just continually decline as far as outcomes in mortality, right? And then, you know, as we start to look at that, it really is incoming of us, particularly in the disparate population research, is that we need to figure this out. So, you know, we'll talk a little bit today about the role of ancestry versus race. And then how do we define that, and how do we have controlled populations and really create a rigorous scientific platform around disparate research and how we translate that into clinical practice. But it starts with us understanding and appreciating the African diaspora and understanding ancestry as a whole. So I like to put this slide up before I get into any biological, because the disparities is a big word, right? So it's a big word, it can mean different things to different people, but they all interrelate, right? So they're all connected, right? You know, healthcare access, health behaviors, you know, there's an epidemiological factor. In fact, you know, many of the early disparity studies were around epidemiological factors, awareness, susceptibility, et cetera. Ethnicity and race, these are social constructs. I think they're colliding now. So a great paper just came out of JAMA last week, talked about how race now is a biological construct, particularly in the context of health, because we see race influencing health, and but that's also a taboo word for example, in society. So starting to understand as the science is starting to grow and mature more, you know, we have to tackle these topics, particularly as we're relating to our patients and our understanding and sensitivity to people that we serve, but also environmental exposures, lifestyles, obviously genetics, right? Understanding the genetic background of that, but all of this goes back into, you know, healthcare equality, equity, and available treatment options, right, for people with diverse backgrounds and as well. So, you know, many of you again are familiar with the NCI Precision Medicine trial, where, you know, the match study where, you know, NCI, I think it was around 10,000 patients. We're trying, you know, basically enroll genome sequence, all the patients, and try to match therapy. And I'm not going to go through the success. There was a number of successes in here, but I like to highlight what didn't occur was that there were a number of patients that we didn't have therapeutics for, right? And so we didn't understand or appreciate the diversity of the genomic landscape in the greater population, and that caused a need for us to go back into the biology and start back and bring new drugs back through the pipeline. So our group has been focused on, and we started off early on, focus on cell signaling, particularly associated with growth factor signaling, epithelial growth factor receptor, EGF receptor. So a group here at JCO showed, you know, very nicely that EGF receptor was overexpressed in African American patients, at least 6-fold. And then our group published a number of papers showing that EGF receptor was a driver, particularly in African-American associated tumors of progressionness, and that led to downstream signaling cascades such as RAS/RAF kinase, that was driving some of the aggressiveness in disease, particularly in late stage disease. And then early on in our career, when I first started lab, we were showing that if you inhibit this, you can actually reverse that, right? So these things are reversible, right? So targeting EGF receptor reversed that, aggressive phenotype, and we were able to see more cohesiveness in the cell population. So, that started us to thinking, okay, well, we have transcription, we have genetics, we have regulation, but there has to be some epigenetic regulation if we can transition cells, phenotypes, and so forth by targeting or inhibiting signaling pathways. So we started looking at methylation, or how the role of DNA methylation or epigenetic roles play in tumor progression. And so again, DNA methylation, just a little background, really important, associated with development, tumor progression, linked to race and lifestyle. The central dogma in DNA methylation is that, if the DNA is unmethylated, it's open, ready for transcriptional access and control. But if it's methylated, it's coiled, and then you get repression of gene silencing. But what actually occurs is that there are a number of methyl binding proteins that actually go and sit on those methyl groups, and they control whether that chromatin is accessible or not, or open or closed, right? And so it's actually, people think methylation is just one step. It's actually a two-step process that's controlled by one of these methyl binding proteins. And so our lab has been focused on one of those, particularly named Kaizo. And so when we first started this some time back, we were really interested in this because Kaizo has bimodal activity. One is a bona fide transcription factor, but it also binds and regulates, sits on these methylated DNA sequences. And that at the time, people were seeing it in the cytoplasm and the nucleus, and no one really understood how that was regulated in the context of tumor progression. So if you kind of think about it in an active type of way, is that if you have methyl groups, again, methyl groups can come from many factors we mentioned earlier, lifestyle, risk factors, diet, etc. There has to be these methyl binding proteins that come and attach to that, and then transcription is repressed. So Jackie Jones was a graduate student. Now she's tenured faculty, actually the first African-American tenured faculty at Troy University. When she was in the lab, she started to ask the question, what was the expression of Kaizo in prostate cancer? And we can start to see, if you look through the progression of disease, particularly if you start here, the sort of low-grade tumors, and sorry, my pointer is not reflecting on the LED screen, but you can start to see it's largely expressed in the cytoplasm. When you switch from low-grade to high-grade tumors, it switches to the nucleus. And what we found is that if you look at here in prostate cancer cells, this is the case of D145, they have largely cytoplasmic Kaizo expression, that if you can treat those cells with EGF, you can actually push it to the nucleus, right? So you can push it in the nucleus. Again, Kaizo is a transcription factor, so being in the nucleus is really important. And if you look at this in disease, particularly associated, we see it increase with tumor progression. And at the time, again, this was kind of back in 2012, early on, disparities, particularly the molecular associated or genetics genes associated with disparities, wasn't as common, but we were sensitive enough to actually diversify our population, our cohort, and we saw that this Kaizo was greatly overexpressed in African-American patients. So we went on to show that it's not just in prostate cancer, so we see it in breast cancer as well. So if you look at, again, even a larger population of patients here, we see the same continuum, even in primary tumors. And if you look at paired mets, Kaizo expressed. And if you look at African-American women who have Kaizo expression, that's equal to European American women who have metastases, and African-American women have the highest level. And Julia Daniels, who's actually cloned and discovered Kaizo when she was a postdoc in Albert Reynolds' lab at Vanderbilt, but now she's at McMaster. She went ahead and did some staining of Kaizo in Barbadian as well as Nigerian population, and we can see even more overexpressed in those patients as well. So you can sort of see Caucasian, African-American, Barbados, as well as Nigeria, women, the progression of the increase. So we feel Kaizo is a bona fide disparity associated marker. We went on to show that it's not just, again, in breast and prostate, pancreatic disease, same thing. And I'm not gonna, we published this some time ago, but just the summary is that we worked out a signaling mechanism where, again, Kaizo was being regulated through EGF receptor signaling, associated with RAS kinase. But at the end of that, control of that ultimate signaling cascade is epigenetic regulation of major important genes such as NF-kappa-B, TNF-alpha, and a number of inflammatory response genes that we know that are associated with progression of disease. If you kind of look at the molecular, sort of the evidence that supports this, if you take, again, these are back to prostate cancer, if you take these prostate cancer cells and you knock down Kaizo, or you treat with 5-AZA, which is, again, a demethylation agent, you can see the same thing. So treating with 5-AZA or knocking down Kaizo, we get the same alleviation of repressed gene expression that we see, and then you see the cells not only transition from this mesenchymal sort of phenotype, which we know is associated with aggressive metastatic disease, then more to that epithelial colloidal-like phenotype as well, and we see that's a direct regulation. And if you go back into the tumors, you see the same thing, right? So early stage EK positive tumors here, which are more cohesive, have low levels of Kaizo, and EK negative tumors have less expression as well. Again, if you knock out Kaizo and you inject it in mice, you get smaller tumors, you get a significant decrease in tumor volume. But what was really remarkable, and I'll show some breast data later, is that 100% of the mice never form metastasis, no mets, right? So we a lot of mice, okay? And we never could see a met in this as well. So really very, very important. And we also published another paper showing that Kaizo is not only regulating gene expression, regulates microRNA expression as well. And you can see that reversal of expression in the histology as well. So this work was done by Bala Karaman, who was a postdoc in the lab and now is a faculty member, and Shana Hardy, who just finished a fellowship at the FDA. So we're really proud of how it progressed. So just in summary of the Kaizo work, this is a little bit early on. It's really important for some of the later things we'll talk about, is that, Kaizo seems to be really regulated through multiple factors. Julia Daniels' lab, you can see here with some of her trainees, worked out we published this paper last year. It was a nice review article that they were working on the TGF-beta signaling pathway through SMADs, so that regulates Kaizo expression as far as in promoting disease. We were very focused on multiple manuscripts, focused on the EGF receptor side, showing EGF receptor, but we know these are definitely co-regulators of EMT metastases and so forth. And so as we start to unravel this, we feel Kaizo is a bona fide target. In fact, we've started molecule development for targeted therapy for patients with high Kaizo expression. So, as we started to think about now, signaling pathways and gene expression, and again, this was some early work on, I started to collaborate with Isaac Powell at Wayne State, who again is a urologist there. And Isaac was asking the question again, back in the days when people were doing microarrays, right? Again, being at Wayne State, they see a large number of African-American in the Detroit area patients. And so he was able to look at about 656 patients there in a very limited microarray. But what he saw was, when he looked at the gene expression, he saw that there were a number of genes differentially expressed in African Americans compared to European American patients, right? And I'm just going to highlight several of them because it's really important. One was ERG. We know tempus ERG is very important in progression of disease, and African American tumors uniformly like ERG and actually like tempus ERG translocalizations. He also saw that interleukins, IL-6, IL-1 beta, all of the inflammatory like markers were also elevated in African Americans. And so again, this work has now been followed up. Renee Reams at Florida A&M. In fact, she actually published a smaller paper before Isaac, but she showed, again, in the Florida cohort, very similar findings that these chemokines, this inflammatory gene signature in African American patients. Stefan Alms, who now has longstanding collaborations. In fact, that's how I met Isaac, through Stefan. Stefan introduced us. We've been collaborating for some time, but Stefan was able to follow this up in a nice cancer research manuscript showing, again, even in an NCI Maryland cohort, that some of the major differences that we see in biology, at least associated with tumor biology between African American and Caucasian tumors, is associated with immune signatures. And so we went back to Isaac's data and we wanted to ask a question. So, we've been working with Isaac for some time, Isaac Powell, that is. and Karamos. And so we said, okay, how is the influence? So ERG is such a major factor now in terms of transfusions, translocalizations. How does that affect inflammatory response in African American versus Caucasians? And then how does that affect what we all, drivers of tumor, of prostate tumors, which is androgen receptor expression? So we started to segment ERG expression. So this is a little bit of complicated graph. So Jason White here is our bioinformaticist. He made this. Bioinformaticists like to make complicated figures, but they're pretty. So if you look at on the left side would be low ERG expression, right? And so if you sort of median, walk that through to the right, which would be high ERG expression, in the middle would be median low, median high ERG expression. And if you look at IL-6 axis versus androgen receptor, what you can see is that in the African American patients, they have low ERG expression. There's high levels of interleukin. That's what I showed. So we confirmed that data. But they also have low androgen receptor expression, right? And so if you segment that by ERG expression, that flips, that changes, and it changes both in the European as well as African Americans. But it's definitely more pronounced in the African American population, right? And so we kind of came up with a sort of theory here is that we see African American patients come in with low, AR expression, particularly in the ERG negative patients, and these patients have high IL-6, and they have this chronic inflammatory sort of signature. This was actually followed up by Tim Rabeck and Schaefer's group in a JCI report, where they showed, they're calling this triple negative prostate cancer, was ERG fusion negative, SPINK positive, SPINK mutations as well. But what they saw is very similar to what we found. So if you look at, a number of patients, particularly in the fusion in negative patients, you can see African American patients have much lower ERG expression, right? But they also have lower androgen receptor expression as well, right? So, again, starting to understand there is clearly some biological driving things, factors that are driving tumor genesis differently. Our lab was really focused on, okay, we're really going to figure this out. We got to have models. We have to have some models to go in the lab and start to dissect this. So early on in our career, Shanice Theodore, again, who is now at the CDC, she set out to establish, in collaboration with John Rim from Walter Reed Hospital, African American cell culture models. So we established several pairs of these models now. In fact, there are a number of manuscripts, and I could just go on. I'm just highlighting some of the major collaborations. We have now, collaborated with numerous labs, and these are actually some of the go-to standard cells that most people use for disparity models that in the field. And so again, understanding that, Lene Woods, who was at City of Hope, and actually it was at Loma Linda when we did this work, we performed ancestry analysis on this, and we can see that our cell lines have more than 70% African ancestry. If you look at the gene expression signature, we also were able to see that at genomic level. So, I'm not sure the familiarity with the disparate cell culture models, but this was really important because at the time, there were a couple of models that were being said to be from African American patients. They weren't really from African ancestry. It was a huge controversy. A lot of papers had to be retracted. And so we needed to verify that in ourselves before we released that to make sure that they were representative of African American and that people were doing and studies were being replicated appropriately. We also had Rick Kittles, again, Rick is at the City of Hope. He performed informative ancestry markers, and he independently validated our cell culture models. Is that not only are these on the side, all of the African American cells, again, breast and prostate and colorectal, also you can see they group here on the right side. And then you can see actually that was the controversial cell line, the E006A H1 HT cells were designated as African American, but they were really European ancestry. In fact, I had a lot of press call me. It was a huge issue. Okay, like I said, it was a huge issue. But now it's sort of like I said, a lot of papers were tracked, a lot of grants had. It was a big deal. So our cell models are verified, validated African ancestry, so we feel appropriate for using them. But that really had us dive into this role of ancestry, right? Because it's important, right? It's starting to become accessible. Again, Rick's lab, pioneered the use of DNA ancestry informative markers, SNPs. But now with with genomics, we can actually do that from sequencing data. And so if you start to look at it, this is how we look at every patient that we start to analyze. So we do global ancestry analysis. So if you start to look at what was strictly done, you have superpopulations, right? African, European, Asian, East Asian, and ag mixed population, and that represents some of the major groups. But now we're actually able to go deeper and do subpopulation analysis as well, okay? So we can actually break up the African ancestry into West Africa versus East Africa, right? And then we actually can derive if those patients are Afro-Caribbean, right? Or they designated as Southwest African. And actually now we are able to even get tribal ancestry analysis as well. And as more and more data comes in, and I'll show you some sequencing data from our patients before, we're going to able to go deeper and deeper to sparse out differences. And so, kind of looking at this from two cohorts, what we thought was really could help validate this. One, this is a southern cohort from our collaborators at UAB, where we took patients, we separated, we did superpopulation analysis, and you look at our European patients versus our African-American patients. And again, you can see, these are African-Americans who self-report as African-American, who present in the clinic, but we see majority of those patients do have predominantly African ancestry, except for one patient here in the middle who reported as African-American, but they actually have East Asian ancestry. That's the one I'm pointing here, all right? But then if you do subpopulation analysis, you can actually show that the European patients actually have ancestry from Utah, right? And that the African-American patients actually have what we call designated as West African ancestry of Southwest USA, right? And so if you start to delineate that, it's not just African ancestry, we can actually go a little bit deeper. And if you follow those patients even further, right, and use several other SNP analysis, those patients who designated as Southwest Americans, they actually have Afro-Caribbean ancestry, right? And those patients from the Barbados, right? And then we were actually able to trace those Afro-Caribbean patients back to their West African ancestry, to the Bantu tribe, and as well, and we see some of those patients have Tuskegee as well. We did this in collaboration also with Isaac. Isaac was very nice to share some of his data with us. And so we actually perform African ancestry on those patients as well. This is really interesting because, again, we see a predominant number of African ancestry, but if we start to look at the European patients here, we can see a range of African ancestry as well. And then we see African Americans also. We see a mixture of Bantu, Yoruba, and then if you actually take that data and you overlay that African ancestry with, the gene signatures, those immune cell population or the immune signature pops up right again. And because Isaac was also able to do whole exome sequencing as well, we see that actually overlap with a unique mutation signature in these patients as well. So if you see here, and I'm sorry, this is a little bit small, but we see these patients have high percentage of SPOP mutations, right? And we see SPOP, a greater percent of African Americans present with SPOP mutation, AR, as well as EGF receptor mutations. And we do see appreciable PT and PI3 kinase, which we know is associated with mutation signature. But we don't see it. We see a complete different reversal of Europeans if we start to separate that out by ancestry. So again, we feel like this is really important as we start to, we don't have a CLIA certified clinical pipeline yet, but it's coming, right? So that we feel like this is really important as we start to look at the patient as a whole. And so just to summarize this piece of it, if you start to look at people who present in the US, right, and whether it's West African ancestry or any ancestry, now with genomics and genomic screening is becoming more and more utilized, we're able to do that right in the clinic really quickly. And we're able to trace that ancestry. In this case, we're focused on African Americans through the Caribbean, through the transatlantic slave trade, and back to tribal populations in Nigeria. We're able to look at a patient as a whole, right? So people, we're all, particularly in the US, admixtures. So we have 70, 80% West African ancestry, what tribe we're from, but also the European ancestry. And what is that European ancestry breakup as well. But what we're seeing is particularly associated with the patients from African ancestry is that these, SPOP, AR, FOXA1, PTM mutations seem to be associated with this immune signature and drive and driving the disease. This is something we need to explore much further, and I'm looking forward to diving into this, but now we're starting to be able to appreciate it because we have the tools to do so. So, you know, again, it's talking about ancestry. It's like, okay, well, you know, how do we really, you know, what is our control population? So we're scientists, right? So you can do ancestry estimates, analysis, but is this really, you know, associated with people from West, that are Native Africans? So in collaboration with Falaki Odedna, who actually just moved to Mayo Clinic, we co-direct the Prostate Cancer Transatlantic program that has about 16 hospitals in throughout West Africa, where we do a number of activities, including outreach, the facility development, as well as we try to get some science in as well. These are just some of the collaborators that we have throughout Nigeria as well as Ghana, and you know, again, a number of the PIs there. There's a really a lot of enthusiasm for the work that we're doing and how we're translating our work that we do in the US actually to improve standard of care as well as influence the research infrastructure in West Africa. And so again, these are some just major collaborators throughout West Africa. I'm actually really excited. I'm not sure if you're familiar with, but Ironman is an advanced international registry to focus on trying to improve the outcomes of men with prostate cancer, where we're doing surveys from a provider perspective as well as a patient, and then collecting serial samples along the way. As a part of our consortium, we actually just opened our Nigerian, COVID delayed us a little bit, but we just opened 2 sites in Nigeria. And again, we talk about a cruise in African-American or people, you know, of African ancestry. In the 1 month, Nigeria has out-collected any sites internationally as well in our first month opening, so the Ironman folks are really excited. And actually, I serve on the diversity working group for the entire Ironman protocol here with a number of people, you know, Dan George at Duke, Elizabeth Heath at Karamos as well, and Franklin Wong, who I mentioned earlier. And we published our first paper on our recruiter strategy to diversify the African-American or diversify the African-American as well as Latino X population in this registry. So it's really important. So utilizing the Ironman infrastructure, we were able to collect samples, right? And so again, we started, you know, genome sequencing. So we did whole exome sequencing again, perform ancestry analysis. And as you can expect, majority of them have, predominantly West African ancestry. But if you compare that to the TCGA, we really see that hold true in African-American patients as well. But this was really interesting too, because we did a PCA plot, right? And so again, Europeans in blue, and these are African-Americans in purple. And if you look at the Nigerian population, these patients from West Africa, although African-Americans cluster more with the West African population, there's actually a pretty good diversity in Nigeria. And so we put these, we colored the Nigerian patients because, you know, we collected from northern Nigeria, middle as well as southern Nigeria. And you can actually see a diversity in their gene expression, well, these are SNPs. That's how we do the ancestry calling. We can actually see a divergence in their population based on whether the patients presented in the northern versus the south, right? And so when we start talking with our collaborators in Nigeria, they say, yeah, well, that's the European influence that you see being closer to the European in the north that you don't see in the south. So again, starting to understand and understand our patient better is really important. So again, in collaboration with Stefan Alms, we actually collected serum patients on this, and these are not just from Nigeria, these are from Ghana. And this time we did, almost, 1,500 patients here, where we looked at African, African-American, and European-American, and we did something called OLinks. OLinks is almost like a Luminex serum analysis. And again, not just in the gene expression, but we saw the immune signature, we see that actually expressed in the serum, and that is one of the major drivers that we see associated with West African ancestry, patients from West Africa, as well as African-American, and then European. And just some of the major drivers that we pulled out was CXCL1, CXCL5, but there, as you can see, it was a really clear gene signature associated, again, in a hierarchical fashion from patients with that are Native Africans. If you actually look at this and you correlate that with outcomes, right? Outcome is the best biomarker that we can have. And again, we break out not just the suppressive immunity score, but if you look at other immune scores such as vascular disease, autophagy, apoptosis, chemotaxis, The one that really is associated with patient mortality, again, is that suppressive immune signature score. And again, one of the major ones we pulled out, again, was one of the TNF alpha ones that associated with a worse outcome. Sorry, that's wrong. And so now that we saw, okay, we replicated, what we see in our African American patients, we see it in serum. What is the genomic landscape, in these patients as well? So again, we perform whole exome sequencing on our Nigerian patients here. And again, what we saw, which was in our Nigerian patients, is high BRCA mutations, right? High APC, ATM. We see this over prevailing DNA damage response signature in African Americans, very high. As we saw in the data from Isaac, SPOC was really high in African Americans. This is from the TCGA, right? And much higher than European American patients as well, right? So again, we're starting to see that there is some ancestral association, and that this DNA damage, even or SPOC, appear to be reoccurring themes that are associated with variants in African American disease. And again, if you sort of pull that out, just looking at BRCA2 or ATM or even APC, we see that, again, really high. And we see that, and not only do we see common mutations, we actually see variants of unknown significance. I'm gonna talk about this in a minute when we get to germline, because we always get asked, okay, call for somatic mutation. What's in the germline? What is prevailing, and this is not just in prostate cancer. What we see now is we're starting to diversify our cohorts, is that when we start to look at the diversity across, I think not just in Africa or across the world, we're starting to get what we call variants of unknown significance, right? Because we're using different references, and so we don't know what they are. And there's a tremendous amount of effort now or interest in trying to go back and recharacterize this or even reformat our baseline genome, because they may not be variants of unknown significance based on the population, right? So again, a huge amount of area of work that has to be done here. But if you kind of look here in the Nigerian patients, again, these are germline mutations. Again, we see almost 100%. This is only 11 patients. We only have 11 true normals, right? We all know it's hard to get a real normal, but these are real true normal patients. 100% of those patients have BRCA1 germline mutation, right? Almost all of those patients were variants, BRCA, variants of unknown significance. And although we don't see a lot in prostate cancer, but if you look at other tumors like breast cancer, this is widely known that, we see these mutations, but we haven't mapped them to a pathological or driver variant. But we also see that occur in others as well. And again, that's higher in the TCGA samples. But that's, again, reversed in European Americans, right? So that's kind of reversed in the germline expression. So in European Americans from the TCGA. So, our lab has been focused on just really trying to find and understand this. And so, just looking through the literature, we also see that prostate cancer clearly has disparities, but we see this immune signature also occur in breast cancer patients. So this is a nice review from Kim et al. That was published in Frontiers Oncology, where they summarize basically what we're seeing in prostate cancer patients, that patients from Africa have this high inflammatory gene signature, right? And they have high, interleukin-6. We showed that in our interferon gamma, and that's associated with a high Treg, tumor suppressor T cells, as well as M2 tumor-associated macrophages. And, again, this is associated with the disparity outcome. So again, breast cancer, just like prostate cancer, subtyping is really important. We have to understand, really dig into deeply to understand how we're going to treat the patients with the right therapeutic. And so again, because of our work in prostate cancer, we started to ask the same thing in breast cancer patients. So we looked at androgen receptor. In fact, androgen receptor seems to be also very important, at least the lack thereof, in breast cancer patients as well. If you look at patients that have negative, or particularly now triple negative breast cancer patients that actually have the lack of androgen receptor, they do worse, actually, as well as we again saw in our temperature negative prostate cancer patients. And if you look at that over, a multitude of papers, this is over 1,000 patients, we see the same reoccurring. These African American patients come in that are AR negative, have a basal-like phenotype, and they do worse, and they have worse outcomes. So in collaboration with Wendy D. Cologne, who was at Tulane at the time, but now has moved to Piedmont Healthcare Center, she's a medical oncologist. We asked, okay, what is the expression of AR in African American women? Again, African American women particularly have over 81% of their tumors are AR and receptor negative. right? So that's an overwhelming percent. And if you look at the association or lack of AR, we see this basal-like phenotype. And I'm not showing the data here, but we see this in prostate as well. So our African-American patients present with this basal-like phenotype associated with this inflammatory type of immune signature. And if you again look at the progression of the disease, these African-American women with basal-like phenotype, they progress faster. So they, Curtis Pettaway, he actually emailed me. He's, chair of urology at Indiana. When I was first starting out, when I was asking our urologist, what do you see in the clinic? What do you see? He said, Clayton, my patients progress fast. My African-American patients skip steps. They skip steps. And so this really informed of how we approach this. And again, we see this in African-American women as well. The time to progression is much faster, particularly associated with these basal-like features. And again, if you start to look at that and we subtype those patients, what we overwhelmingly see, particularly in the African-American, we're calling these quadruple negative, not triple negative, quadruple negative, because they lack AR as well. And we published this quite nicely. But these quadruple negative patients actually have this basal-like signature, right? And that seems to be associated with the driver of disease. We validated this in combination with CARES molecular profile. I'm sure you guys could familiar with CARES, where we looked at from a multi-institutional perspective over 870 patients. Over 79% of patients that were so-called triple negative are actually quadruple negative, right? So this seems to be one of the major phenotypes. And those patients who were metastatic or recurrent, 75% of those patients also were AR negative. And so again, if you look at from a biopsy perspective, and this is a really cool slide, but it's a little complicated, so I'm gonna walk you through it. So what I designated as the top row would be what the patient, the phenotype or the characteristics of the patient at the first biopsy, and the second, which is a circle diagram, will be what was at the second biopsy if the patient either was recurrent or metastatic. Okay, so if a patient presented with a luminal A phenotype at the second biopsy, if that patient was either recurrent or metastatic, that 70% of those patients retained the luminal A. But I'll fast forward to the real focus here is that if that patient presented as triple negative, only 50% of those patients stayed triple negative, right? But if that patient was initially quadruple negative, 75% of those patients stayed quadruple negative, whether they were recurrent or not. So we think the lack of AR is actually even a better diagnostic marker, particularly in breast cancer patients. So just diving into this a little bit further, again, back to our ancestry analysis in collaboration with Melissa Davis at Cornell, as well as Lisa Newman, who's a surgeon, and Upenda Manning, who's at UAB. How we did ancestry analysis on African-American women. And then if we start to overlay again African ancestry, right, with self-reported race, and you take self-reported Europeans and your African-Americans on the right, you actually start to see European Americans who self-reported as European Americans who have a little bit predominantly more West African ancestry actually share more gene signatures with African Americans. Right? And if you look at that phylogenetically, you can start to see that branch off. There's a branch of European Americans with this predominant African ancestry that don't cluster with Europeans that have more European ancestry, right? So this starts to become really important. So again, really wanted to see if we could replicate this immune signature. Shakir Ahmed, who was a postdoc at NIH, when he was in the lab, we did enrichment for gene immune signatures, and we see again the classic MAP kinase and kappa B chemokine signaling associated with these diseases associated with African-American breast cancer tumors. We wanted to go back to, okay, what is the role of Kaiser? How do we understand the molecular drivers of this? So we looked at our Kaiser expression in our cell lines, and again, this is all over the place, a very busy slide. But this is the really important piece, is that when we took our Kaiser knockout model and we overlaid that with our patient population looking for what we so-called Kaiser driver signatures, what we pulled out was TGF-beta1, which we know is really important, as well as CD47 and FKB. And CD47, we were really interested in, so we started to follow that up. So again, looking at our Kaiser knockout model in our breast cancer cells, same thing in prostate, we see no metastases and lower tumor growth. But we also start to see a reversal of CD47 and the Kaiser knockout, SERPA alpha, and as well as tumor suppressor gene thrombospondin 1. And if you start to look at these tumors in a mouse that lacks Kaiser, we see a more infiltration of M1 macrophages, which are more favorable, the phagocytic versus the suppressive macrophage markers, which is CD206 expression. And if we go back in the lab and we take exosomes or conditioned media from our kaiser knockout model, and we treat those exosomes and cells from kaiser knockout versus control, we can actually influence macrophage polarization. So you see these macrophages actually polarized toward an M1 in the lab versus a classical M2, which we know is associated with disease. And then those cells, the exosomes taken from cells that have black kaiser, they actually are myophagocytic. So they're actually phagocytosing, actively phagocytosing cancer cells compared to the suppressive environment that we see when patients in tumor cell lines that have high kaiser. So again, just to bring this back to from a patient perspective, it's like, okay, we see this phenomenon in the lab. Okay, what does this mean in the clinic? So in collaboration with Kevin Gardner, who's Vice Chair of Pathology at Columbia, we went back and did multispectral imaging on a number of patients where we wanted to look at the localization, the co-expression or correlation of kaiser with macrophage infiltration as well as CD8 infiltration as well, or PD-L1. And again, what we saw here, this is a little bit of a busy slide, but the sort of take home here is that patients that have high kaiser expression have higher infiltration of CD8 cells, but those CD8 cells also express PD-1, right? So they have this sort of exhaustion signature. And then we also see macrophage infiltration actually closer in a race-dependent manner to these CD8 positive PD-L1 or PD-1 positive, PD-L1 positive CD8 T cells, right? So if you start to think about, from our cell biology work, is that kaiser is creating an environment that sort of mediating the suppressive activity, all right, or suppression of the T cell activity. So again, just a little bit of summary of these findings is that we feel that, in this case, exosomes, but starting off with tumors that have high kaiser, we see high expression of macrophage markers such as PD-L1. This influences differentiation of macrophages. So if you have high, you differentiate more to the M2. M2 is associated, macrophage is associated with increase in markers such as SERPA alpha, CD47, PD-L1. That's how cells escape immunity. If you have low kaiser, it's associated with more of that M1 macrophage phenotype. And then that's how you have active immunity, right? So you have these macrophages and alleviation of suppressed T cell activity as well. So again, this is really important because CD47 has just ended, well, not just ended, now in phase 2, but it's a number of companies, Big Pharma has introduced a number of CD47 antibodies in clinic. I think they're in phase 2, particularly for multiple cancer types. And this is really important as we're starting to understand the biology that we can translate that, right? So these are real world trials going on, and we have some strong evidence that, we should start accruing African American patients in these trials as well. But just from our perspective, and we mentioned before, is that we were really interested in therapeutic. We saw this macrophage, the role of the macrophage is a real targetable event, right? Something that we could really target and modulate and have a significant impact on suppressed immune environments. So again, just a sort of a summary is that, Th1 responses, which is active immunity, Th2, which is suppressed immunity, macrophages are right there at the center of it. I like to say we all live in a T cell world, but there are other influences of T cell activity, right? And so if you think about macrophages, M1 macrophages being good and M2 macrophages being bad and associated with progressive disease, the goal is to increase these M1 macrophage phenotype. So in collaboration with Jesse James, who's a biochemist at Tuskegee, and we actually collaborate with Udo Ulof at the NCI, we actually created or developed a macrophage inhibitor targeting a CD206 that's really highly expressed on M2 macrophages. We did single X-ray analysis where we were able to do the first three-dimensional structure of that. And this molecule actually binds and targets CD206. And through a folding activity that we were able to see here through negative stain electron microscopy, we actually see that folding of, this is actually not a small molecule, it's a peptide inhibitor. That peptide binds to the CD2 receptor. It actually folds and initiates the signaling cascade. And so again, the goal here is that we see that our macrophage inhibitor, the targeting, which is a canonical marker of actually M2 macrophages, CD206, we target that receptor and we're able to rapidly transition these M2 macrophages back into the M1 phenotype. And we see that not only in cancer, but we see it in fibrosis. And in fact, the first clinical trial is going to be fibrosis. fibrosis, and scleroderma, as well as kidney and lung fibrosis. And so again, just treating with this, it happens almost de novo, right? So even as early as 30 minutes, we can treat, CD206 high, CD86 low expressing macrophage. We start to see a shift in populations, and you actually, here on the right, you can start to see these CD206 cells after 24 hours, they have this gradual shift over to that M1 phenotype. We published this really nicely. It's over 150 figures in this manuscript in Science Translational Medicine last year, where binding to this initiates a complex signaling cascade, which ends up activating NF-kappa B, which then actually causes a transient release of TNF-alpha, which initiates this active immunity we see with cooperation with T-cell activation. But in the autogrammatic, it signals back to the macrophages to cause CD206 macrophages to actually undergo apoptosis. And again, we see, decreased expression in PD-1, PD-L1, and SERPA alpha that we saw earlier. This really drives the disease. And then we look at this in mice, and again, this is P16, KRAS mice. If you treat it with gemcitabine, I know Braxane is standard of care now, but gem was at the time. You can start to see a combination of gem plus our peptide really suppress tumor expression. And not only suppress tumor expression, we see a decrease in the macrophages. But and again, I'm sorry that my pointer is not, but again, you can see a increase in CD8 expression, as well as infiltration, and a decrease in T regs, right? So we start to see that reverse phenotype as well. If you look at it as a combination with PD-L1, very similar, right? So we get responders and non-responders, mice, we all get that. But again, if you co-treat with our peptide, suppressing the M2 macrophage phenotype, we get all responders, right? We see a net benefit. And we see that codependent on the T-cell infiltration, right? So if you actually co-treat those patients, these patients, these are P16 mice, with CD8 antibody, you can mitigate that response, right? So there's a macrophage response that has to be elicited, but that macrophage response turns into a net T-cell response that cooperatively influences the overall activity of the immune microenvironment. And we just published another paper with Roger Lowe's lab at UCLA, where he saw the very same thing. So Roger Lowe was really interested in MAP kinase inhibitors. We know MAP kinase inhibitors, have recurrence. They have fast recurrence. But if you actually think about, so what Roger was in his lab was thinking about, if you start to look at the time of recurrence, right, and at the time of recurrence, what is the population of cells that actually mitigate that? And when they did CyTOF and single cell analysis on that, they saw again it was these macrophages. Macrophages popped up, and the macrophages, particularly the tumor-associated macrophage phenotype. So he actually he reached out to us and he said, hey, you guys got this peptide. We saw the paper in Science. Can we actually co-treat our tumors that are actually recurring with your peptide to see if we can sustain activity? So it's a little bit of complicated animal experiment, but what we did was we treated with our CD206 target peptide, co-treated with the MAP kinase inhibitor, right? And these are actually P53 BRAF melanoma mutant mice, and we did this in KRAS mutants and BRAF mutant as well, so it holds true in several mutational types. You're able to sustain activity of the MAP kinase inhibitor. And not only is sustained activity of the MAP kinase inhibitor, these mice actually don't respond to PD-L1 either. And now they actually respond to PD-L1. So you're able to sustain the MAP kinase inhibitor and respond to PD-L1 by the addition of suppressing the macrophage population through our peptide CD206. So again, I kind of bring this back to our disparity, right? So, if we start thinking about immune therapy, and how we translate this, we really feel like we're sort of bringing it full circle, right? And so Oliver Sorter, who is a colleague as well, we've done some work with, I think he performed one of the pivotal clinical trials, and again, this is Provenge, but when he looked at his African American patients and he treated them with Provenge therapy, what he saw was that African American patients did 2 times better on Provenge than European American patients, right? And they reported that at ASCO, I think 2 years ago, right? So again, this is proof of principle that we were headed in the right direction. Actually, I showed Oliver sort of our data. He was like, "Keep going. This is really cool." So we were again encouraged that we were headed in the right direction to not only, provide solution for people, particularly in prostate cancer patients that don't typically respond to immune checkpoint inhibitors, that we can get them to respond, but also African-American patients or diverse populations that typically have to suppress immune environment. And so we started thinking about moving forward, like, what is the future, right? How do we sort of bring this all together? Integrated omics workflows, I think, are going to be the key of how we move forward. Technology is allowing us to be able to capture that, capture not only the mutational signature, but the tumor microenvironment. Again, this is being able to take samples, RNA, DNA, protein, serum. We've demonstrated that we have the ability to do that, have that coordinatively influence, that have those samples available, performing multi-level omics, right? And I have genomics, epigenomics, transcriptomics. What I didn't have here, we should, is the demographics, right? We think demographics, social demographics is really important. I'm calling it the sociome. We're going to add it to our omics because we need to understand that patient at every level in order to accurately be available to treat them. And then apply, cutting edge technology, machine learning, deep learning algorithms, right? Genome sequencing. We're pioneering a single cell analysis on FFP samples called CLIA Codex. We're doing that actually in collaboration with Kim Bleeman here at Yale. We have a mark proposal in where we're comparing IMC Fluidigm, IMC single cell analysis to a CLIA. Fingers crossed that the mark grants gets funded. Understanding that at the level, what technologies are best appropriate to be able to understand the diversity of the cell population in our patients is really, really critical. But then forming bioinformatics teams and workflows, and not just for research, clinical, CLIA certified bioinformatics workflows that we can rapidly understand the phenotypes and genotypes of our patients, and then translate back that for many of you guys who can inform how you make decisions for your patients on a daily basis. This work takes a ton of collaboration and kind of work, a lot of money as well. So I'm very thankful for a number of funding sources and a number of long-standing collaborations that we've had. I think diversity starts with diverse thinking, and it starts with diverse people. So I like to keep a diverse lab of gender diversity as well as race and ethnicity as well. And I'm really excited because these guys do all the work. I just go out and talk about it. So thank you very much. And,
questions? It's fantastic.
I have a question. So that was your absolutely sensational talk, really a tour de force of that topic. So many questions come to mind. I guess the first, I was intrigued by the data on Ancestry, and it makes me, wonder: are there blind spots in the way that we clinically get race? Some of the prognostic gene signatures we were, essentially validated in Caucasian and European ancestry populations, and when they've done studies to compare the differences in expression, maybe they're looking at is it possible they're looking at the wrong targets? Is there, do you think, a role of incorporating ancestry into clinical decision making?
We think so. Yeah, that's really the point of this. That's the take home. So again, it starts in the lab, but really excited. Some of this mark proposal led by Cornell. That's what we're trying to do. So we're trying to do a proof of principle study. Take that patient, we do CLIA, whole genome sequencing. This is going to be done at the New York Genome Center, right? But they have clinical workflows, but trying to add ancestry in almost immediately, understand those ancestry informative markers, and if we see different mutations, send that back to the provider really immediately, but also send that back to more in-depth staining and analysis so that we can see that real-time influence maybe the treatment strategy. So we see that. Some people think it's a little further away, but I don't think it's too far off that we would be able to do ancestry informative analysis in real time in a CLIA certified workflow and have that available in a report.
Phenomenal. Yeah. And my other question was about the role of modifiable factors. I think that there's some data looking at the differences of social determinants of health. you look, for example, in the VA population, a lot of the racial disparities compared to Serum Medicare or other populations appear to be attenuated. Right? So some of that relates to timeliness of care, equal access, and factors like that. So I don't know how to, it's obviously no simple answer how to account for that. But I wonder if context has been, if you thought about looking at context, treatment context, is a patient been treated in a facility where they've had equally timed diagnoses?
Yeah, so we yes and no, we thought about it, and I didn't show the data here. But in our breast cancer patient, it was more pronounced than our prostate cancer. So we actually had, remember, I think mentioned there was some people that self-reported as one ancestry was different. We actually went back and actually looked at genes associated with self-reported race versus ancestry. They're different. So we started looking at self-reported ancestry. We started seeing stuff like diabetes, cardiovascular disease, other comorbidities. We look at ancestry genes, we saw things more like immunity. So again, we think that's why I think it's really important, because you start to understand that. So you can start to pull out, okay, these could be modifiable, right? Because these are not genetic. So we could modify this if it's a self-reported demographic issue, it's an access to care issue, it's an environmental lifestyle issue. We can tease these out. These may be more genetic, where, you know, I'm a basic scientist, we need to develop targeted therapies, you know, or vice versa. So we see that as the future. You hit it, the head of the nail. that's the future. Absolutely.
Morning, this is Dan Petrolak. Quick question. Great talk. In this issue about the androgen receptor and the loss of the androgen receptor, extrapolating that, one would expect that there would also be a higher rate of neuroendocrine or small cell carcinoma. Is that something that you've observed or experienced with these patients?
You asked a really good question. We don't have enough patients that we collect. So you know, the neuroendocrine phenotype, you know, from what I've seen, it mostly is ADT induced, right? So we don't see that patient population enough in the clinical trials to be able to see that. So, but when we've looked at the AR negative patients, we don't necessarily see the neuroendocrine phenotype. We see this basal phenotype, and we see this infiltration of immune response, but it's not necessarily neuroendocrine before treatment, at least data so far.
It's interesting because there does seem to be two phenotypes. One, of course, the de novo, which may be biologically different than the acquired, as you had mentioned. So it'd be interesting to see if that de novo phenotype is expressed at a higher rate, you know, because of this observation that you made. I think that would be a nice little correlate to look at.
Yeah, absolutely. You know, we just haven't had enough patients to tell us, but I think you hit the head on the nail, because, you know, obviously the loss of AR, you know, prostate cancer bearing neuroendocrine phenotype is definitely, you know, a real critical, factor. And actually there's a lot of interest around that these days of people trying to understand that. So absolutely.
Other questions?
I did. Sorry. So my question has to do with the samples that were collected from like Nigeria, et cetera. Is there any plans to collect samples from the Caribbean? I'm just curious about that population in comparison to that of Nigeria or that of Africa.
Yeah, so absolutely. So we actually have a sister consortium called AC3, so the African Caribbean Consortium. And so they do all of the, so they're actually opening the Ironman in Caribbean.
Okay.
So we work together. So NCI said, okay, you guys are in West Africa, Nigeria. So they actually said, we're not going to fund you both. You guys got to work together. so we work together. You know, NCI makes so we actually work together in tandem, but they handle the Afro-Caribbean population. That's headed by Camille Reagan, who's at Fox Chase, right? But again, we share data. They actually did some of the genome sequencing. We did some of their analysis. I'm just not showing it here. But we did some of the analysis in their patient population as well. So yeah, similar. Yeah.
I have a last question. That was a fantastic talk. Now, you're thinking about more aggressive phenotypes and tumors that evolve. Have you thought about taking it to the next level and focusing more on the more aggressive aspects, comparing the metastatic lesions to tumors that have evolved from a local lesion to a metastatic one, and compare that difference in ancestry in a metastatic lesion? Because those are ones that are becoming more aggressive, and what makes them more aggressive in some subpopulations.
That was the grant that just got announced this week, our PCF award.
I just know you're talking, I'm sorry.
Yeah, no, I have no data, we have a little bit of data, but we just were awarded a PCF grant with the team. We're looking at metastatic samples, local to metastatic samples. The goal is to do actually single cell sequencing and cell-free DNA analysis on those patients from metastatic lesions and compare that just as you, like you said. So November and PCF funded us this week, it was announced Tuesday. So yeah, we got to get started on it. But absolutely. So yeah, hit on that. Absolutely.
Congratulations on the grant.
Well, thank you.
All right, well, absolutely fantastic. Thank you.